want to introduce the next speakers. And the next speaker in the series on November 15th is Peter Singer. And Peter is going to change his title to The Suffering of Wild Animals. Should we do anything about it? And if so, what? And last talk of the semester on our stone on December 6th. And the title is Small Scale Dynamics with Environmental Implications. Today, Steve Pacala, who is the Frederick Petrie Professor of Ecology and Evolutionary Biology, will talk on the topic of, the title is Steve.
Competition on water.
Competition in water, okay.
I have a lot to say, so make this fast.
Steve got his degree from Dartmouth and Stanford. He's a very active person. He's the chair and I think founder actually of Climate Central. It's a non-profit dedicated to the education of the public on climate change and influence of decision makers. He's a member of the board of the Environmental Fund, co-director of the Carbon Competition Initiative at Princeton, and past director of GEI. He got lots and lots and lots of honors. I mentioned the fact that he's a member, he's a very young member, I should say, of the National Academy of Sciences. And among many things I chose too, that was sort of fellow AAAS and so forth, is an honorary member of the British Ecological Society, which seems to be He's also a lifetime fellow of the Ecological Society of America. Steve.
Thank you. Well, it's a real pleasure to see people from lots of different departments and from Climate Central and from GFDL and friends here. I hope I won't disappoint you. This is not a wedges talk. This is a talk about science, all right? It's a talk about relatively recent science, which for me has been exciting because usually old dogs don't get to try to do any new tricks. Now, I'm going to speak unmicrophoned here to Geo 10 because I've done it many times in classes, but if anybody in the back or anywhere else has trouble hearing, just raise your hands, scream, whatever you need to, and I will increase the volume and increase the force of the sound. I'm going to talk about competition hydraulic damage and the universal rules regulating plant water use. Now, at the very beginning, I want to acknowledge two collaborators. This has been a blast because the effort really represents a collaboration among a bunch of different people. Adam Wolf, who's in the audience here, is the CEO of a company called Arable. Here with his pulse pod, he sells a sensor that tells you what makes a plant happy, right, roughly. And this idea is fundamentally Adam's. He came up with it and figured out the answer by sort of intuition well in advance, and I thought it was probably false. So very much this is the case that he had this worked out by some sort of mysterious hardwire process in his head. Bill Anderegg is now an assistant professor at the University of Utah. They were both here as postdocs for a couple of years. Bill has a lot of the supernatural energy necessary to do the empirical tests that I'll get to at the end of the talk. Now, in some ways, this subject, this talk, is painful to me. And it's painful because I taught introductory biology for EEB for about 10 years. And I had one rule about what I was going to present. Everything I said had to be true 10 years after the date of the delivery. All right? I wasn't going to tell anybody anything that was on the cutting edge because it was high probability it was going to be untrue. These people were going to medical school. They had to be able to give me the right answers on exams, that sort of thing. And so this work I'm talking about today, the theoretical paradigm and enormous empirical evidence about what governs plant water use, was part of that, of the group of ideas inside the field that were really well known and well established, and they became a cornerstone of my teaching. I had this wonderful narrative that went from the origin of photosynthesis 3 billion years ago in a world without oxygen, free oxygen around, and how it led to constraints on the biosphere's carbon cycle and everything else later on. The gist of it is still true, but the end point is unfortunately now false in my view. All right? So that's why it's painful. I thought this was well established, and it really had an enormous amount of evidence, but it turns out I now believe it's not true, and that's what this seminar is about. Okay, so the outline for the talk is that it starts with the obligatory nod to the carbon cycle and the important global warming problem that this is part of. I then want to talk about the importance of water in the terrestrial biosphere. I need to give you some background material so that you'll understand the rest of it. I have to talk about the paradigm of 40 years, right? The 40-year paradigm that I taught in introductory biology for years. And then I want to talk about an alternative and a direct empirical test of the two, and then what the implications are. So let's start with the carbon cycle. You know, the thing that we almost always acknowledge in PEI seminars about global warming in the biosphere is that anthropogenic emissions, more than half of them don't end up in the atmosphere but are absorbed by sinks. A little more than half of that half goes into the land biosphere. The land biosphere is gaining weight, right? And half the half goes into the oceans. And so I've been concerned since coming to Princeton 4 years ago now, with understanding and predicting the land biosphere sink. Now, this has come to the fore recently in the Paris Accord. I just want to cue you in if you haven't been paying attention to the Paris Accord. There's originally little discussed Article 4, Section 1, which says that so as to achieve a balance between anthropogenic emissions by sources and removals by sinks of greenhouse gases in the second half of the century. Now, I talked to Todd Stern, the chief negotiator from the American delegation last week, had dinner with him in New York, and he said this was deliberately left ambiguous. But it is a statement about the end of increased greenhouse forcing in the atmosphere. When sources balance sinks, the concentrations don't go up anymore, all right? And a reasonable interpretation is that anthropogenic sources should be balanced by natural plus anthropogenic sinks, beginning about mid-century, which is 33 years from today. That's a pretty tall order. So this is just a cartoon of what that would look like, where in 2050, sinks from the ocean and land balance anthropogenic sources of fossil emissions and other emissions from agriculture and deforestation and that sort of thing. But the important thing here is that the sinks at that point determine the size of the fossil economy. All right, so it hits everyone in the pocketbook. All this is just to reaffirm that sinks are important and understanding sinks is important. So most of the time that I've been at Princeton, virtually every other science talk I've given is about how to develop a model of the terrestrial biosphere. The terrestrial biosphere's carbon uptake, the carbon sinks created by the terrestrial biosphere, in a world in which we have in biology no analog to the Navier-Stokes equations, no fundamental equations that govern the biosphere. And so for most of my time here, for just about every other talk I've ever given, on the carbon cycle, I was talking about that problem. And what we've all done over the last 2.5 decades is to take a class of models that do scaling correctly. Individual-based forest simulators, where you can make a bunch of measurements on trees and predict 100 and 1,000-year dynamics of forest that are used all over the world to manage forests because they work, where analyses of these models actually cause legislatures to pass laws about how the land is going to be managed. They've actually crossed and become mature, crossed from science to engineering, right? Where we've got a set of models that are relatively good, and what the exercise has been is to try to scale those up to the globe. And the most important part of that scale up, and the intellectually most satisfying part, has been to analyze these. This just simply shows you a forest from above, right? The trees are moving around, growing to fill gaps in the canopy. They have phototropism. The trunks move around a bit. This actually shows two species in a forest simulator. And what you do is you throw down a little random box in the stochastic process, wait T years, and say, is there a tree of this species and size in it? And you do that thousands and thousands of times, or you do it analytically. And then if you take the fraction of time that there's a tree of that species and size in the box, divide by the size of the box, then that is the density of trees of that species and size at that time. And then the goal is to derive macroscopic equations that predict the time evolution of that density. So most of my talks have been about that, and I'm not going to talk about that at all. And the reason is that, to some extent, I sort of feel like we can declare victory on that. We have multiple ways of doing this now, and most of the models, including GFDLs and NCARs and the Hadley Center and everybody else, uses the technology that we've developed here over the years to do exactly this sort of thing. And so, with Adam and Bill coming to Princeton, I turned my attention, and my lab has turned its attention to water. Now, I want to talk about then the importance of water in the terrestrial biosphere. By my way of thinking, water was the second most important thing, because wood is the primary carbon in the terrestrial biosphere. If you show me a molecule of carbon in the terrestrial biosphere, more than half of it is or used to be wood. And wood is a weapon that trees use in height-structured competition, and so getting this sort of height-structured competition that was force simulators did well right was at the core, the zeroth order about what we had to do. But if I were a water person to begin with, I would have said that water was the most important thing, and we should have worked first on that. Now, why is that? I've included this graph because this guy, Mike Rosenzweig, was at Princeton, I think for his PhD, right? And in Eno Hall, I suspect he compiled these data in Eno Hall. Comes from 1968. And what this shows is actual evapotranspiration, amount of water leaving a system, versus the primary productivity, the amount of carbon the system brings in annually from photosynthesis from plants and the amount that's left over after respiration. It is a log-log plot, but nonetheless, there's an obvious trend there. The obvious implication is that fundamentally the carbon cycle, this is the size of the engine in the carbon cycle, is determined and constrained by available water. All right? This is another graph that you often see in talks about the terrestrial biosphere. This is the so-called Whittaker diagram from 1970. It has average annual temperature here, plotted to 30 degrees Celsius. These are cold places, minus 15. Average annual precipitation from 0 to 450 centimeters. And what Whittaker did was he took the mean precipitation and the mean temperature from a whole bunch of different locations and said, what does the vegetation look like in those locations? Put dots down, and then drew boxes around similar vegetation, called those biomes. And there are no points out here because it's impossible for really cold places to rain a lot, simply because cold air doesn't hold much moisture. But nonetheless, what you see here is negative diagonal banding of types with sort of similar amounts of carbon above ground. So here's tropical rainforest, temperate rainforest, tropical seasonal forest, temperate forest, taiga. These are all forms of sort of forest. And the negative diagonal banding is because that represents equal amounts of water. When it gets hot and wet, it's the same as when it gets cooler and drier because the evaporation is less. So the amount available to the plants is about the same. And the negative diagonal banding in the Whittaker diagram is fundamentally caused by the water constraint. So what are we going to do about it? Well, in this business, there's been a paradigm that has been in place at least since 1977, with an enormous amount of empirical evidence behind it, and that is used to govern plant water use in all of our Earth system and climate models now. And at the heart of this is a stomate. Now, this graphic image is an electron micrograph of a stomate looked at closely. This is a little pore in a plant leaf. And the thing that you're looking into is the substomatal pore. And the reason these things are so important is that the substomatal pore is surrounded by moist, gooey, wet surfaces. And so the water inside it is saturated with water vapor. And so when this pore opens up, that saturated water vapor wants to escape, and that's where the water loss is coming. Now, the bottom of that substomatal pore is connected to a series of tubes that extend with fine roots throughout the depth of the soil. And so these plants are able to dry the soil from different depths all simultaneously. If they weren't in place, then what would happen, I learned this from Chris Milley ages ago, if you just had a bare soil surface, it would dry from the top, and that dry layer would reduce the amount of evaporation a lot. And this is why, in fact, he discovered a strong relationship between deforestation and runoff, for instance, in the Amazon. All right? So these guys are really critically important in determining how the atmosphere and land surface are coupled. Now, these two cells here, those are two cells, they're guard cells. And you probably remember from introductory biology, if you were a person who was who had to take it, that these things are banana-shaped. And what they do is they increase the concentration of dissolved substances inside their cytoplasm by pumping ions into it and cause themselves to plump up. And because they're banana-shaped, that causes them to open. And if they dump the ions out, that causes them to close. All right? Now, it turns out this is not quite the way it works, but I'll get back to that in a minute. I want to give you a little bit of background to understand these some more. Now, the units that we're going to be using are water potentials. And water potential is the energy necessary to convert water from its current state into a reference state. And the reference state is typically water at 20 degrees Celsius and 1 atmosphere of pressure. Okay? Water potentials in nature are typically negative because water wants to run spontaneously from one to the other. It runs downhill. Very negative water potentials. Plants have very negative water potentials in their xylem, in their pipes. Generally, I'm going to use the word water tension, which is minus water potential. Think of tension as suction. When xylem has high tension, it means it's sucking really hard on the water in the soil. All right, so I'm going to use negative water potential, which is positive water suction, and because I think it's just easier for people to think in positive units. So think about that as suction. Now, this is a micrograph of a substomatal pore, and it's from a paper that's in press in PNAS with Adam, Bill, and me. These are the guard cells. This is the substomatal pore. This is the mesophyll. And this is the terminus of the pipe system that extends into the soil. All right? Now, the important thing is that whatever you learned in introductory biology, the way this really works is that the plant has both extensor muscles, increases in the pressure of the water inside the guard cells, pushes them open by making those banana-shaped things rigid, and increase in the water potential in the epidermal cells plumps them up and squeezes the stomates closed. All right? So it has flexor and extensor muscles just the way we do. It's also amazingly set up so that the epidermal cells are connected directly to the water in the xylem, whereas the guard cells are isolated from that and get their water from the gas phase in the substomatal pore. And this difference allows the plant to balance the flexor and extensor muscles and regulate the opening of the stomates, which runs the large parts of the hydrologic cycle. Now, we worked on this because we wanted to be sure that none of this system was passive. And I'm just going to assert to you now that everything I'm going to say from here out is the result of a deliberate biochemical algorithm inside these little guard cells which runs them. And none of it is passive. If this system were to follow passive forces, everything would work backwards. Stomates would open when it got dry and close when it got wet, rather than the reverse, which is what they do. Okay? So that's the important take-home message here. The relative strengths of the flexors and extensors is regulated by the active adjustment of the concentration of dissolved substances inside the guard cells. Now, another thing that you need to know before we go further is that although biologists often talk about stomates as the only valves operating that are between the soil and the biosphere through the plant. There's actually a second valve. It's a second valve. And it's related to a phenomenon where when the tension gets high and the plants are sucking really hard, embolisms happen. Air bubbles get sucked through the side of the stem and effectively block the xylem elements. These are xylem elements of a badly cavitated stem. The blue blobs are the ones that still have water in them, and the remaining ones are blocked. And so this is called the conductivity. It's the way engineers and scientists measure the capacity of a conduit to have water move through it. You can think of it as the diameter of a hose, right? It's high conductivity is big diameter, low conductivity is low diameter. And the important point here is that the conductivity falls off as water tension goes from low to high. It falls off because the elements become increasingly blocked by air. Now, most plants can refill these the next night. Some can't, but most of them can refill these the next night at some cost. So this is a second shutoff valve, if you want. The rate of flow through a stem is actually the sum of all the little conductivities along the way from the roots up to the terminus of the xylem in the leaf. If you're a mathematician, then what you do is you integrate this conductivity function from one potential to another. And the upshot is that as leaf potential goes from low to high, if you look at the rate of water flow, as tension gets high, the rate of water flow goes up the stem, but at diminishing returns. All right? So it falls off, and it's because as you get up here, the plant is sucking harder and harder and harder, but through an ever smaller pipe because so many of the pipes become blocked. Okay? So that's the second shutoff, is this embolism shutoff from the conductivity. Okay, now I promised Francois that I would have exactly one slide with math in it, and this is it. I'm going to go through it a little bit, but then we're going to reduce it to a blue box, which is never seen again except as an icon. Okay? So what I'm going to need is a set of equations. Now, these are relatively standard. These are equations that are inside just about every Earth system model. Every model of the carbon cycle has these equations. And actually, this is a reduced set. This is a set of equations you'd need to predict carbon uptake and water loss for a leaf under windy conditions. If the conditions aren't windy, we need an energy balance equation and some other stuff here. But for now, just focus on these four. The first equation is an equation that governs photosynthesis. This thing is a light-limited photosynthesis term. That's the incoming flux of photons. That's the amount of light that's shining on it. This is the dark reaction limited. So photosynthesis uses energy from sunlight to create, to trap the energy of sunlight, and then uses that in an enzymatic reaction to force CO2 into a sugar. The first term governs the first part of that, the energy engine from the photon capture. The second part of this governs the second. That's the enzymatic reaction with carbon dioxide. And it's the minimum of the two that determines the photosynthetic rate. This is the leaf respiration rate. This is called the Farquhar photosynthesis model. And the beautiful thing about this is that it actually works. This is for C3 plants, which are the dominant plants that are out there. And this was first invented by the biosphere something around 3 billion years ago. The second equation is Fick's law. It just says that photosynthesis is all about the production, the consumption of CO2 to put it into sugar. So this is a CO2 consumption term. It's the amount of carbon pulled out of the atmosphere around the leaf and it put into sugars. And that sink for carbon must be balanced by carbon that flows in from atmospheric carbon dioxide, that concentration, minus, and the gradient between the atmospheric carbon dioxide and the amount of carbon inside the substomatal pore in the leaf. It's flow down that concentration gradient through the stomate, which is open by this much. GS is the stomatal conductance. That's how open the stomate is. That must balance. The carbon in must balance the carbon out through photosynthesis. The 1.6 is because of a difference between CO2 and water, not important here. Evaporative water loss is simply stomatal conductance, how open the pores are, times D, which is the saturation deficit. It's how dry the air is. That's why it's a D. It's the gap between saturated air and the current air. All right? So that's D. D big means dry air. And finally, this is the integral that I talked about of all the different little pieces of conductivity along the way from the soil to the leaf. And that also has to balance evaporation because the water at the bottom of the plant has to balance the water that leaves at the top of the plant. Now, there are 4 equations here, but there are 5 unknowns. It's the internal concentration of CO2 in a leaf, atmospheric, the photosynthetic rate, evaporative water loss rate, the water potential inside the xylem, and the stomatal conductance. So we need a 5th equation. Now, in the future, these equations are going to be represented by that blue box, and you're just going to see them like that, okay? But understand that these are always there, okay? And actually a more complicated set are always there, just the way they're there in just about every model of the carbon cycle. They're inside GFDL's land surface model. They're just all over the place. And it's because they work. I mean, this is a success story, right? These things work. Now, the 5th equation in all of our climate models comes from an empirical relationship. An empirical relationship that was first discovered by Adam Wolf's thesis advisor, Joe Barry, but then has been applied all over the world. It turns out that there's an empirical equation relating stomatal conductance, how open stomates are, and a bunch of other things. And that this relationship is amazing because everywhere you go, you get the same functional forms all over the place. There is super abundant data from literally hundreds of species from all the biomes of the world. This is amazing, all right? An enormous empirical regularity. The first part of that regularity, and these are just 2 different, this is predicted versus actual from some simple empirical relationships. That's the predicted by a simple statistical model, and that's the actual. And these things all have the same form. First of all, stomatal conductance, how open stomates are, is proportional to the photosynthetic rate. Literally just proportional. Secondly, stomatal conductance is inversely proportional to the atmospheric CO2 minus a constant. So there's a hyperbolic dependence on CO2. And thirdly, the stomatal conductance is inversely proportional. It's hyperbolically dependent on the dryness of the air. Those 3 things, that's it. That's the simple empirical model that underpins all of the stomatal conductance in models. It's proportional to photosynthesis, and it's inversely proportional to linear functions of CO2 and water. Now, that's pretty nifty that we've got so much data. There are a couple of different forms of this, but they're not different in important ways. They're all statistically indistinguishable from one another. The one that's in our model and most models is one by a guy named Leuning. But that's not important for our purposes here. Now, again, the empirical models have enormous support from all over the place. This is one of the most well-supported empirical relationships that I know of in biology, maybe the best. But what guards against the wrong out-of-sample behavior in future climates? The problem with an empirical relationship that you don't understand is that if conditions change, maybe it won't work anymore. All right? Now, I've got a little graphic that shows that just for people who aren't sort of up on this kind of thinking. We've got 3 different data points, and we fit 2 different models to them. One of them has the correct functional form, the red one, and one of them has an incorrect functional form. It's the difference between a quadratic, I think, and a cubic or something in here. And then we've got a new out-of-sample data from sort of more extreme conditions that we never saw before. If we've got the correct functional form, the idea is that we ought to be able to hit the out-of-sample data. And if we've got an incorrect functional form, we might be radically off the way the blue line is from the green dot. Okay? That's all we're talking about here when I talk about what guards against out-of-sample behavior. You fitted things in the current world, now the world's changing. Is it still going to work when you don't understand why the empirical relationship exists? Now, fortunately for this theory, and the reason this thing made the cut for introductory biology for me, was that there in fact is a reason to believe the current empirical functional forms. And this is a 40-year-old paper by Cowan and Graham Farquhar, the same guy who invented that photosynthesis model we were just talking about. And it's called the water use efficiency hypothesis, which I'm going to just abbreviate as the WUEH. Okay? And this was first written down in an obscure journal in 1977. And it's been cited just thousands and thousands and thousands and thousands of times and is universally accepted, including by me, during introductory biology. Now, what does this say? Cowan and Farquhar argued that, if a plant has fixed available water, then its carbon gain is going to be best if it can use that water efficiently. Okay? If it can use the water efficiently. And so they did a little mathematical exercise with what are known as with the constrained optimization problems, and proved that in that case, stomates should be regulated so that the following is true. Stomate is regulated, and you say, now what happens if I open my stomate a little more? Didn't follow the rule, but a little more. Well, I'd get more photosynthesis because it increases the rate at which CO2 comes into the plant, and I'm eating the CO2. It's more food for me. And so you'd get a little increase in the amount of carbon gain from a little increase in stomatal aperture. But you'd also get an increase in water loss because now that thing's open more and more of that humid air is escaping. And so Cowan and Farquhar said this thing should be this times a constant water price lambda. If you were an economist, you'd call this a shadow price. It's the carbon value of water, and that this should be a constant. All right? Think of it as that a plant should be regulated such that the carbon it's leaving on the table, by not opening its stomates even more, should be equal to the amount of water it doesn't want to lose times a constant water price. Okay? That's what the thing says. Now, the water use efficiency hypothesis works for a relatively simple reason, and I think I have time to go through this. If you look at carbon gain as a function of stomatal conductance, it goes up like this. It goes up like this because the more they open, the more CO2 comes in, the more plants have to eat, the more carbon they get to fix. It falls over because this is ultimately enzymatic, and eventually the CO2 fixing enzymes get saturated with CO2. They're open enough and they can't process it fast enough. Evaporative water loss is simply proportional to how open the stomates are. But if the air is dry, they lose more water than if the air is low. Now, this water use efficiency hypothesis criterion says take the slope of the top curve and the slope of the bottom curve and take the ratio, and that's plotted over here. Change in carbon gain over delta over change in stomatal conductance over change in E over change in stomatal conductance, where the changes in stomatal conductance cancel. And you plot those two by just plotting the ratio of those two slopes, and you get two different curves, one for wet air and one for dry air against stomatal conductance. And it says that lambda here, the water price, ought to be constant. And so you can see that the intersection here is for open stomates in wet air and closed stomates in dry air. And the reason for this fundamentally is that with fixed available water, a plant doesn't want to open stomates more in dry air, because if it does so, the amount of carbon it gains per unit water loss is too high. And so it's going to run out of water blowing it on low carbon gain days. And so what it should do instead is save that water below ground for use later when the atmosphere is moister, when it can get the same amount of carbon with less water loss. And the price lambda is just the highest value that you can choose that actually uses all the water available to you. You could set that price wrong and never use all your water, leave some of it below ground. But if you set it just right and you follow this rule, it's the maximum carbon gain you could possibly get. So this makes some sense, but only if the plants can keep water below ground for later use in conditions where they get more carbon gain per unit water loss. Now, you can do the same sort of argument for why it is that the water use efficiency hypothesis explains the qualitative dependence on photosynthetic rate and CO2 concentration that's observed, just the way it does for for the dryness of the air. But there's another way to do this, and there's a giant literature here, and this is really the linchpin of the case for why this body of work became so widely accepted. So if we take the 4 biophysical equations and the water use efficiency hypothesis thing, that delta C gain over delta E ought to be a constant, and work on them mathematically, there are many, many papers going back over a period of over 20 years that show that these together imply the formula that is empirically observed, with all the dependencies, the proportionality of photosynthesis, the inverse proportionality to CO2, the inverse proportionality to saturation deficit, so that they argue this means that empirical rule is an evolutionary optimum, and thus the world is going to behave that way even when it changes. Plants are hardwired to do this, okay? So that's the argument. Now, to recap, the empirical model in all of our GCMs and all of our climate models that regulates plant water use is supported by data from many, many species and biomes, and an evolutionary optimization argument that says it ought to work even out of sample. All of this is good, and that's why I taught this thing, right, all the time. But there's some cracks in the firmament that we've all been ignoring for a long time. The first one, and this is actually kind of minor, but I include it here for completeness, is that there's no generally accepted way to predict lambda, the shadow water price. You can measure it, measure what a plant is using, but you can't predict what it ought to be. And that's weird, especially because we know that it changes over long time scales. This is much more important. The model is missing something fundamental during drought, because it doesn't predict the stomatal closure that happens when the soil dries. This is a review paper from Klein et al. In 2014 that shows some fitted traces to stomatal data. This is the leaf water tension. So the leaf is sucking a lot here and little here. It's under hydraulic stress here and not here. And this is the stomatal conductance. And plants close their stomates when it gets dry, all right? And the simple model doesn't explain any of this. Is not included. Now, as a result, climate models have had to include a completely unconstrained phenomenological function to do this. They call it a beta factor. And what they do is they take the stomatal conductance formula from the water use efficiency hypothesis and multiply it by this beta thing. And the beta thing falls from 1 to 0 as the water potential in the soil, in this case, gets large, okay? And so what this does is it's just a phenomenological switch that closes the plants down that's unsupported by data, all right? All the models include one of these things. Now that's dangerous. Whenever that happens in a model, it leaves the modelers free to start tuning it to compensate for other problems in the model. Now Anna Trugman, who's a graduate student here, and I think she's here somewhere. You can ask her about any questions. You're going to have to talk to her. And Bill Anderegg have a really magical paper that is now submitted for publication, in which they took 9 of the big climate models, and they just asked, how important is this switch in these models? And they had a very simple metric. There are other metrics you could use, but this is very simple. I said, well, if it opened all the way up, how much extra carbon would the biosphere fix? So these things are partway shut, and they measured it all over the world from runs that were archived of these models. And so this is the extra amount of carbon that would be fixed if that switch were opened. Now this model would store an extra 240 billion metric tons of carbon. Now I want to emphasize that total anthropogenic production is 10, not 240. The largest term in the carbon cycle currently is GPP, the total amount of carbon fixed by photosynthesis. On land, it's about 120, and then in the oceans, it's about 120. And this is as big as that. This model, in contrast, has 20, not 240. So this is the danger. Intermodel variation is double GPP, and it shows the danger. This is embarrassing, okay? It's embarrassing for the community. You've got something unconstrained. You've had to add it to make it work. But you know, by looking at this, that this is going to be a disaster when we're predicting the response of the biosphere to drought. This model is going to behave very differently from this model, All right? This has got to get cleaned up. So I was unaware that I think all of us were, and that's why Anna's paper is so important. But this is going to be one of the primary uncertainties that govern the predictions of models into the future. I bet it's the single biggest one. That's my guess. Now, the third crack in the firmament is that fundamentally, the water use efficiency hypothesis optimization only makes sense if plants can save water below ground. All right? But the problem is that there's superabundant evidence that show that plants are thieves that steal each other's water. All right? So if you leave it below ground, what's to keep your neighbor from taking it? All right? First of all, this is a guy steaming the soil off of high-pressure steam to steam the soil off roots. And people have done this all over the place in forests, and typically a tree's roots extend for 3 times its crown radius, so encompasses 10 neighbors or so. Secondly, there's a lot of studies now using molecular markers. You go to a place, you grab some fine roots, you PCR them up, you sequence them, and you figure out how many different species are there. There's typically 10 in any different given root sample. Many individuals competing for the same little spot. And thirdly, Adam Wolf had a Grand Challenges study that was really kind of magical because he added isotopically labeled water to one tree and shows that half of it comes out of the tree's neighbors. All right, so it's a very direct way of doing this in the pine barrens. Okay, so plants do steal each other's water. Now, those of you who are as old as I am may remember Danny Cohen, who used to visit here. Danny is a wonderful person, a theoretician from Israel. And he, from a very early time, actually before the water use efficiency hypothesis was first written down, was arguing that it's nonsense to expect a plant to store water below ground because other plants will steal it and they'll outcompete you because they do better. Right? They take your water and use it for themselves. They'll overtop you. They'll take all your light. And so this is his quote from an article that says effectively that the only way the water use efficiency hypothesis could be an evolutionary optimum is if the plants cooperated not to steal each other's water. It's formally the solution of a cooperative game, not a competitive game. And so we were really interested in looking at the competitive game, and I'll get to that in a minute. The fourth crack in the firmament is that a lot of work had developed subsequent to the water use efficiency hypothesis being written down that shows that there are extra costs of low xylem potentials. There's the cost of refilling the embolized vessels. There's the reduced photosynthesis due to partial desiccation of leaves. There's reduced future photosynthesis when your xylem is damaged and can't bring stuff up from the bottom. All of these costs, I'm going to define later, the sum of them, as this little function here, theta as a function of psi L. As theta gets big, the costs are, so this measures the costs. As water tension gets big, costs go up. And this is the sum of all these different costs. So we expect this function to be concave up because damage gets progressive and costs should go up as water tension gets up. And I'm going to be talking about the current cost per unit of tension, which is minus the derivative of this function. The negative comes from the fact that tension is minus water potential. Okay? And so this is the cost per unit tension, which is the slope of that curve. All right? And we expect that to be rising also if this curve is concave up. If that curve were linear, then we'd expect this thing to be flat. All right? All right, so an alternative hypothesis. Adam Wolf, Bill Anderegg, and I started to work on the competitive alternative to the cooperative game that led to the water use efficiency hypothesis, right? And so we added below ground water that the plants were sharing, so they were able to steal each other's water. We added the costs of xylem damage to the model, and we also made an assumption for this first go-round that is false in general, that costs at one time don't influence costs at another. All right, so that the costs are independent at different times. It's a much more difficult but still important problem to look at the non-independent cases. And then we went to work on those 4 biophysical equations and the rest of it and said, what is the evolutionary stable strategy? Evolutionary stable strategy is jargon invented by a guy named John Maynard Smith to talk about the solution of a competitive game. And at an evolutionary stable strategy, it has the following characteristics. If you're a player and you play a strategy that's the ESS, any other strategy can't invade you, can't increase when rare against you. If somebody else has a strategy that is ubiquitous, and you're a rare invader with the ESS strategy, you can invade against them. All right? Now, John Nash is the person who sort of formalized this as an idea, and it's why one of the reasons he won the Nobel Prize. One of my fondest memories at Princeton is the first time a couple decades ago when Nash wandered into my office, because I have a full floor-to-ceiling blackboard in there, and just started to derive something on it. So I watched him for a while, and he turned out to do this repeatedly over a period of months. He would just like wander in and start to do math, and I just kind of watched, try to figure it out. And then every once in a while he'd ask me some question that always seemed unrelated, but that he was interested in and was related to what he was thinking about. The first time he said, "You know anything about chimpanzees?" And the answer is, I do know something about chimpanzees, and we talked about it. It was really, okay, so this is all a tribute to John Nash. All right? And we went to work on this and discovered that the ESS stomatal algorithm doesn't do this. It, in fact, simply maximizes carbon gain instantaneously. Plants should regulate their stomates so that they just make the most possible. They don't care how much water they use. They should just gobble up as much as they need to maximize their carbon gain. They're voracious. They don't plan for the future. All right? They just get whatever they want right now that makes them have the highest carbon gain. And so we took the 4 biophysical equations and this optimization criterion, which we call the carbon maximization hypothesis, and this one with the water use efficiency hypothesis. And this summer, all this work from here on out is done since July 1st. Okay? Solve for the optimal stomatal algorithm from this big mess, which is actually a little bit tricky. And the amazing thing happened. I would have bet $1 bazillion that this wouldn't happen. What happened was that both of these hypotheses predicted the observed responses that have all the empirical support actually equally well. They predict identical functional forms, no difference between the two. I would have bet a lot of money that didn't happen, but it did. But they both also predict the observed beta shutoff and the formula for the observed beta shutoff. But the beta shutoffs predicted are different. This is the conductivity function. This is the beta function predicted by the carbon maximization hypothesis, and this by the old water use efficiency hypothesis. The important point is they're different. When you've got the carbon maximization hypothesis, the optimum is for the plants to shut their stomates before they damage their xylem. That's what that graph shows. All right? Bill Landeregg went and measured these things for a whole bunch of different species. This is a measure of how of the potential at which conductivity is half shut off, and this is a measure of how much the potential at which the tension at which the stomates are half shut, and the dots below the 1-to-1 line shows that prediction is in fact correct. Plants do shut their stomates before they damage their xylem. So to recap, the carbon maximization hypothesis gives the optimal stomatal algorithm with competition, the water use hypothesis without a water use efficiency hypothesis without competition. They both predict the observed responses of stomates to the CO2 and photosynthesis and water vapor, which were used to justify the water use efficiency hypothesis equally well. But the carbon maximization hypothesis predicts a beta function closer to what is observed in nature. And now I want to end the talk with a discussion of a direct test of the 2 different optimization criteria. All right? Now, Bill Landeregg and Adam and I have a paper now on this, which has been submitted to Science. I think it's pretty good, but I expect some wagon circling in the reviewing community because people have dedicated their lives to the water use efficiency hypothesis, and as you'll see, I think we have an extremely strong falsification of it. So it hinges on this thing called the marginal xylem tension efficiency. The marginal xylem tension efficiency just says, if I open my stomates a little more, how much extra carbon would I gain by extra CO2 coming in over how much my leaf tension would increase, how much my xylem tension would increase. So think of this as a measure of how much carbon I'm leaving on the table by not opening my stomates any further in order to avoid an increase in the tension inside my xylem that's damaging me. All right? That's all this thing is. And we showed that the formula for the MXTE, this thing, is identical to the optimization criteria in each respective case. So when you test the functional forms of the MXTEs, you are testing the optimization criteria themselves. They're the same thing, just expressed in a slightly different form. The water use efficiency MXTE here is a function of two terms. This term is really from the water use efficiency hypothesis. It says, this is the carbon that I leave on the table by not opening my stomates a little further in order to avoid the extra water I'm going to lose. All right? Because that's what that's all about, with the water price. It's proportional to how much extra water you're going to lose. And this term is about how much damage that tension is going to do to your photosynthetic system and how much carbon that's going to cost you. And that's proportional to how low the tension is, how big the tension is inside your xylem, not how much water you lose. Now, the critical difference is that this term declines as tension goes up. It declines as tension goes up simply because as the tension goes up, the pipes get blocked more and more with air bubbles. And so the water flow that you get is ever diminishing as the tension goes down. So that so what this term is doing is saying, I'm leaving photosynthesis on the table because I don't want to lose extra water. But as the tension goes up, you lose less and less water for each increment in tension because the pipe becomes more and more blocked with air bubbles. So this part of the MXT from this term, from the water use efficiency hypothesis, should be decreasing, whereas this one's always increasing. Okay? Also, the carbon maximization MXTE is just the second term. So here's the critical thing. The water use efficiency hypothesis MXTE should have a decreasing component. The CMH MXTE should have only increasing components. And so the CMH MXTEs can have these forms, the water use efficiency MXTEs can have those forms, and you can simply test this against data. And that's what we did in this paper. Bill used his contacts to track down all of the data that was suitable for this. That turns out to be a lot harder than you would think because you need all of the empirical drivers of those 4 biophysical equations to do it. And in particular, you need to know the soil water potential. And the way to do that is you have to get up before dawn to measure the xylem potential inside a plant because it equilibrates with the soil overnight. And plant ecologists don't like to get up before dawn. If they wanted to get up before dawn, they would be bird biologists, okay? And so I really think that's what it is, all right? I think that's the difference. And anyway, so they eventually came up with 34 species where we had enough data. And there are a bunch of different outcomes that are possible. And I'm not going to go through this in detail. This is a proper statistical test. There's all kinds of statistics. There's p-values. There's multiple test corrections. There's all this stuff. I'll talk to any of you. I'll make you sick with talk about this if you let me. All right. In these two cases, the water use efficiency hypothesis is right. And my little moniker for this is that plants plan ahead and control their appetites. They don't eat all the time because they're saving themselves for when conditions are good for losing water. These two cases, which are just different, are that plants live in the moment and have no self-control. So when there's not enough data, you can't tell that either of these could be true. Then I'm saying these are like non-voters. This is a lot like Bernie Sanders. He probably does believe that plants cooperate, right? Or would if you just took away lousy federal regulations. Hillary likes to have it all, right? So this is both water use efficiency hypothesis, but with some hydraulic costs. At the other end, of course, right, Trump. It's like, damages accelerate, doesn't care. Just, I want to stuff it in my mouth, all right? And finally, this is Paul Ryan. he's like the little Donald, hopes the damages aren't going to get too bad, right? So all right. So what was the answer to these statistical tests? Well, the first thing is the data came from really all the different biomes. And the outcome, if you look at species, was a shutout. There were only a few, 3, where we didn't have enough data to say anything. For all the ones where we had enough data, 32, the CMH was supported and not the water use efficiency hypothesis. If you look at the total number of days over which we had enough data, same thing. It's a shutout, all right? So this is as clean a statistical result as I've ever seen, all right? And this isn't close. If you actually look at the statistical support, the CMH hypotheses are many orders of magnitude more likely. If you look at a posterior probability density after confronting the models with data, it's many, many orders of magnitude. The posterior supports the CMH. The final thing is that when you do this, you have to estimate this theta prime, because the beta function we have from the CMH is just a simple function of k, which we already had in theta prime. We now have a constraint on this unconstrained part of models. Okay, so the conclusions. The data overwhelmingly support the CMH instead of the water use efficiency hypothesis. The CMH is consistent with competition. The water use efficiency hypothesis isn't. They identically predict CO2 VPD and photosynthetic rate dependence of stomatal conductance, which has been the primary evidence, empirical evidence, supporting the water use efficiency hypothesis. The CMH also predicts that. But the CMH also predicts the observed stomatal closure during drought. And finally, we have estimates finally to constrain this most unconstrained parts of models, which should improve our prediction, should improve our performance of our system models. And finally, 10 years of teaching about plant water use and EB is all wrong. And if you're interested as an alum and want a refund, you should contact Lars Hedin, okay? He'll talk to you. Don't contact me, all right? So I'd like to thank Adam and Bill for having all the energy and brilliance to think this up in advance. Funding agencies, and to all of you for 24 years now plus of a lot of fun. Thanks. Kind of long. Yeah. Sorry.
For those who want to stay, a few minutes for questions.
Yeah. Yeah, Suki. I'll repeat this.
Some plant, C4 or C3 plant, some plant
for terrestrial plants. There's C3 photosynthesis, which is the majority. That's 3 billion years old. There's C4 photosynthesis, which was invented by evolution about, 20 million years ago? Yeah, several times, and it's in phytoplankton. There's many independent, and C4 photosynthesis is interesting because it actually has a different model, right? Different model for the photosynthetic equations. Interestingly, it has the same empirical stomatal model. We've not done this work for the C4 photosynthesis equations, but it's an interesting and important thing to do. We got this far by the end of the summer, and now I'm swamped, right? But we will do it. So I don't know the answer to your question. The third kind is cam plants. Cam plants are like cacti that during the night open their stomates and grab CO2, and then during the day use that stored CO2. So they have a different strategy. They aren't governed by these equations, but they're a very small part of the biosphere as a whole. Okay. Yeah, Sean. Yeah, so as you know, we've worked a lot on the ESS allocations of photosynthate to roots and fine roots and stems and leaves and rooting depth and all that sort of stuff. And so the plant is obviously a mix of a whole bunch of different optimization criteria. The interesting thing, though, is that our ESS stomatal algorithm is orthogonal to that. So so I guarantee you that it'll still be the ESS no matter what the outcome of that evolutionary game. Okay. Yeah.
What about seed dispersal? Because you can imagine if all of your neighbors are your children, you have less likely to steal from them than
Yeah, so that's called kin selection, and it's really an important topic. I think that kin selection in plants is something that hasn't really been thought about very hard. I mean, if you think about wood, I often say that wood is a weapon to kill your neighbor and her children. But the flip side is you don't want to kill your own, and they're oftentimes clustered around you. So kin selection is important, and it's at least possible that you could imagine a kin-selected game that would lead to cooperative water use. But when we've tried to fiddle around with this, look at local dispersal and look at precisely, Caroline Ferrier, who's a former grad student of mine, who's now first year as an assistant professor at the University of Texas, has actually worked that problem a little bit. And the effects of competition on the outcome are a slippery slope. So she finds that if there's any large-scale penetration of roots from neighbors that aren't your children at all, the thing then runs away and slides all the way to nasty, right? You lose all the cooperation quickly. And it's because somebody who doesn't, well, you can think about why that is, right? Damaging the other guy's children is more important than damaging your own. Yes?
Are some plant species more vulnerable to xylem damage than others?
Some plant species are more susceptible to xylem damage than others. I have two new water interested postdocs in my lab, Megan Bartlett and Matteo Detto, and I are talking about actually, Megan has discovered an axis that is a trait axis that looks like it separates plants that care a lot about water and that are easily damaged from plants that are armored up and don't care so much. And their stomatal algorithms reflect that. And what we're talking about doing is estimating where on that continuum all the different plant species in this Panama rainforest in BCI are. And then Matteo has 4 years of eddy covariance tower evaporation flux data from that tower. I think we'll be able to see a signature of the diversity of strategies, the diversity of trees on the biosphere-atmosphere coupling. And if correct, we may actually have an impact of biodiversity on the climate itself, which would be really interesting. So we're actually out to quantify that, but technically it's very challenging to do. Yes. Lena.
One of the often criticized that they don't represent biodiversity at all, like plant and functional plant, hydropower, food and water. If you would have to improve this data function, how important it is to understand differences among the different species to capture right response to drought and to the warming?
So that's part of this Panama project. I'd like to actually look at the cloud of diversity for all the traits that affect water that are out there, run like a dimensional reduction, like a principal components analysis on it, and then examine in models how much of that matters. So if you were to replace the diversity of it, say 10 or 15 or 200 types, right, along some set of axes with their midpoint, what difference does it make? That's precisely what we want to do. And not just in a model, but against large-scale data. All right, so we're out to answer that question, and the answer is I don't know. My intuition says you want to count like a physicist, 1, 2, 3, infinity, right? And we currently count up to 10, which is probably a bad idea. 4 through 10 is a waste. Yeah. Yeah, so there's a hydraulic lift problem where it's possible, for instance, when the atmosphere, so one of the interesting phenomena is when the atmosphere is moist at night, or relatively moist, low vapor pressure deficit because the temperature goes down and the soil is dry, if you leave your stomates open, you can actually get water moving from the atmosphere down through the plant, out your fine roots, and into the soil directly around your root tips. And then when the sun comes back up, that reverses and you get to suck up that water that you'd gotten during the night. So that's an interesting wrinkle. So the answer is I don't know. My guess is that it'll turn out to be okay, just because I think competition rules the day, but I'd be lying if I said we'd analyze that case. Yes, I think hydraulic lift is real interesting. I also think capacitance is interesting. The fact that stems swell at night as they come into equilibrium with the water in the soil and then shrinks. They have some stored water. That water may not be subjected to the normal competition rules because they already captured it. Maybe that water they want to use prudently. So the question is, one of the ways one of the paradoxes about the evolution of plants was that terrestrial plants didn't evolve really till about 400 million years ago. So what was going on for the other 3 billion years where there was terrestrial but no plant? All right, well, were they lazy? And something fundamental had to be developed. And there are a couple ideas about this. One is that the xylem elements themselves are the critical piece. And the second is that a symbiotic relationship between fungi and green algae, one that provided the roots and the other that provided the photosynthesis, was what was necessary to develop, and that was hard. So since that time, I've just thought about fungi as fancy plant roots. But it would be interesting to usually I think about the fungal symbionts as being much more important for nitrogen and phosphorus acquisition than for water acquisition. But there is evidence that they do influence water acquisition too. So the simple answer is I don't know, and it's a good thing to look at.
Thank you, Steve.
Thank you.